Alrighty, welcome back. We were talking a little bit beforehand. I am in an Ireland jersey, and Professor Elaine O'Reilly is a professor at University of Dublin. Is it that the university official name?
University College
Hopefully that's
Dublin.
University College Dublin. University College Dublin?
Yeah.
That doesn't really exist in the States. That's kind of funny. So firstly, I guess I doubt many people are going to know anything about Ireland and Dublin. So, I know you kind of grew up. You're from that area. Did you grow up from the farmlands in Ireland, or are you mainly from Dublin for your entire life?
Yeah. No, I'm born and bred in Dublin, and did my all my schooling and things here as well. So I didn't go too far from home for my early part of my education. I stayed really close to mum and dad and the comforts of the home environment. But yeah, it's a great place to live. It's a great place to study, and it's a great place to visit.
I definitely. We were talking a little before. I'll definitely be over in Ireland through the coming years. I guess some other questions I had about it though is that I feel like a lot of people are obviously going to know about Dublin. Like that's kind of like the first thing that at least Americans, right? It's like, oh, Dublin, Ireland, but they probably don't really know much about other than like I would say the rich history and also other places to go see in Ireland. If I know this correct, I think Dublin was actually founded by like Vikings, or like the very first Nordic people kind of established Ireland as like a trading post, I think. And then eventually, basically, the Irish people kind of took it back over eventually, but like I think it was established by the Vikings. So um, I don't know how true
Yeah,
that is
yeah,
though. So I don't know
you're
if you want
going to
to
know
enlighten us on
more.
that.
You're going to know more Irish history. You'll know more Irish history than I will probably, so you'll quickly catch up on me there. But yeah, that's absolutely a really deep Viking heritage here as well. But Irish history is a complex matter, and we've been taken over by many people and clawed it back in the past. But that's a different podcast and a different story. But yeah, really rich heritage there. I
Yeah,
mean, and you mentioned where to visit in Ireland. Dublin is fantastic, but actually there are so many places to visit in Ireland. You could spend a long time here, and there's just really beautiful places to visit right throughout the country.
yeah, it's quite a beautiful country. Really unfortunate though. I don't know if the Irish women national teams unfortunately didn't make it out of the group stage, but it was really cool to see them in the tournament for the Women's World Cup. So hopefully they come back stronger next. Either I guess for the upcoming Euros would be Women's Euros would be 2 years from now, I think. But hopefully they come back stronger for the next World Cup, and I guess it'd be 2027. Um, but anyway, as you mentioned, you studied at University College Dublin for your um, undergraduate in chemistry. I'm kind of curious, is like was STEM something that you're always interested in, or kind of when did that journey start for you?
Yeah, when I was in school, I wasn't a particularly great student. I enjoyed other things in life other than my studies, but I was always okay. But I had a reasonable interest in biology. I was always quite interested in that. And actually, I went to UCD with the vision that I was going to study genetics. I was really interested in Darwinian
Mm,
evolution and in all aspects of genetics. And that was my plan, but of course, plans change. And I suppose as a student going through, it was a general science program in UCD. I really liked the chemistry, the school of chemistry. It was a really welcoming environment. I really loved the lectures, and that kind of just sucked me in. I didn't even do chemistry for my leaving certificate, which is the high school exam that you know your final exams. I didn't even study chemistry for that, so it was all new to me. You quickly pick up something if you're keen on it, and that's it. It was really the environment and the people that led me down that path for my undergraduate degree. And then I guess after that, you in Ireland and in UCD, you do a stage 4 research project in the final year of your
mm.
undergraduate, and that kind of got me interested in research.
Yeah. So what did you yeah, did you do like any undergraduate research at UCD at the time, or was it mostly just lectures?
Yeah, so for your first, second, and third year of the degree, it's mostly lectures, but you have a huge lab-based component as well. So you start off in first year in undergraduate laboratories, but as you can imagine, it's all quite following a recipe, and it's not really research. It's more learning the techniques and learning the skills, which is absolutely important. And then in your fourth year, you join a research group. In my case, actually, back when I was a student, you joined two different research groups, sort of split in over half the year. And that was really where my love for research came, because for the first time, you don't really have the answers. The experiment isn't necessarily expected to work. It's actual research, and you really see what the life of a PhD student is like. And it really appealed to me. So that's where I got sucked into the PhD.
Yeah, so you did your PhD at UCD as well. You joined Professor Francesca Paradisi. And
That's
so,
right. Yes.
what was about her lab? What were you doing in that lab? And what were some like your favorite memories as a graduate student?
Yeah. So when I was an undergraduate student in my final year, Francesca Paradisi, who's now a professor at the University of Bern, she just joined UCD as a young academic, and she came in to give us a bit of an introductory lecture, introduce herself and say hello to us as final year students. And it was a real breath of fresh air for me, because as an undergraduate student, whilst I loved chemistry, I loved organic chemistry probably mostly, I had still a real passion for the more biological side of science and research. And Francesca's research seemed to be very much a mix of that. So she was using enzymes to do organic chemistry. And immediately I thought, yes, this is what I've been looking for. So I approached Francesca and said that I was really keen on research, and would she agree to take me on as a PhD student? And she agreed, and we applied for some funding, and we got that. And it was really one of the best experiences of my life so far. My PhD, as lots of people will say, was just such a good time. You've got lots of freedom to explore different ideas. You're with usually a great bunch of people, and I was with a fantastic bunch of people in a great institution. And it was just brilliant. Obviously hard work and sometimes stressful, as PhDs can be, but a really all-round fantastic experience.
Yeah, like anything in life, everything that's like difficult, like definitely does not come easy. I guess what I'm curious is, I feel like I said this before, but I feel like there's such a unique opportunity to work with a newly professor, because obviously they're trying to get tenure, so they're guaranteed at least 5-6 years to get that. And so you have a unique opportunity where if you're joining that first year, you're like in some ways in the trenches with them. And so it's kind of a really unique experience that really only certain students get with that that first year. So that's really interesting. Yeah. What was your PhD thesis on? And so I know you guys use enzymes to do organic synthesis, but I'm sure there's a lot of different enzymes. I'm sure there's obviously a lot of different reactions you guys do. So how do you guys focus on
So
which reactions?
my PhD with Francesca was an unusual experience, in the sense that what I set out to do, maybe it's not that unusual actually, what I set out to do in my thesis, which was to use a lipase actually to make biodiesel. Biodiesel was really topical back then, and we were trying to take just waste vegetable oil and use a lipase to make biodiesel from that. But what we set out to do just didn't work out well at all. And actually, I switched about a year and a half into my PhD. I went very stressed into my supervisor and said, this isn't working. I'm not enjoying this very much. And I just want to do some synthetic chemistry. And she was a fantastic mentor and a very reasonable human being. And she said, sure, what would you like to do? And so I actually spent the next 18 months making non-proteinogenic amino acids, which had some biological application, and we developed some methodology to make those. So I know it's not uncommon for the first year or maybe more of somebody's PhD to just be not a waste of time. It certainly is never that, but that it doesn't end up really being a major part of your final thesis. And I was lucky to have a supervisor which really let me, sort of follow my own interests, and she had enough confidence in me that she let me sort of change my mind and change gears quite a bit. And it worked out very well, actually. And just to mention that the first 18 months there, was absolutely crucial in the next steps of my career, which led me on to a postdoc, because what I learned in that first 18 months, although it didn't really end up being a key part of my PhD thesis, the skills and the ability to work with enzymes to produce proteins really helped me out in the next, well, up until now, the rest of my career.
Yeah. I think it's super important for people to hear, especially like young graduate students. Like you said, like 18 months, not like definitely like not down the drain by any means. The research didn't work out, but certainly the skills that you picked up are, I mean, you can't even put a price on how valuable they are. I think that's super important to hear, especially when you're working with enzymes. Like I don't know a thing about like preparation and handling enzymes, so I don't know. I don't know how difficult it is. Like I don't know like what the procedures are. So actually, it'd be pretty interesting to talk about. Actually, why are they so difficult to work with? Like what makes them so I don't know hard.
Yeah, so not all of them are. Some of them are very straightforward to work with, and they behave really well. So producing these enzymes from DNA, and you have a DNA template, which you essentially, typically, you ask a cell, it might be a microbial cell, like a bacterial cell, you actually ask that bacterial cell to produce the protein, which of course is the enzyme that you're interested in. And the molecular machinery of that cell is, of course, already geared up to produce these proteins and these enzymes. So that technology has been developed over many years and it works very well. And some of them express, as in you can produce the protein very easily. Some of them don't. And if you think about on a very basic level why that might be, if you're using something like a bacterial cell and you're using the molecular machinery of that cell to produce the protein from your gene, that's okay if the gene is easily produced in that bacteria. So maybe if it's another bacterial enzyme, but if you're trying to produce a mammalian enzyme, so a human enzyme or even a plant enzyme, which come from organisms which are more complex than bacteria, sometimes the molecular machinery within that cell means that production of that protein is just not as straightforward, and the environment that it's produced in is just not suitable. There are other reasons, but that's one of the reasons. But sometimes they're very easy to work with, and I think a misconception sometimes by communities, academic community who don't work with enzymes, say synthetic chemists, is that it's really difficult to work with them, and there's some sort of it's a dark box and we don't really know what's happening. I mean, that's not true anymore. In fact, we have really well developed toolboxes of biocatalysts that you can purchase from a company and throw into your reaction as a powder, and it's just like using any other catalyst. So we've come a really long way even in the past 15 years since I started being active in this area.
Yeah, I when we touch on what you research doing, like what you're doing in research now, I definitely like was curious about how, like, I don't know how, like, let's say efficient, let's say adding like enzymes into a chemical reaction. So we'll definitely touch on that in a little bit though. But yeah, I think it's super interesting. It definitely puts a new paradigm on how we think about chemical synthesis for sure. Because yeah, I mean, nature really is the best at these things, so we definitely have a lot to learn from it. Just how accurate and even just like an anti-selective these enzymes can be. It's crazy. It's crazy to think about, and so we definitely have a lot to learn from it. Now, at the end of your PhD, you went to go do a postdoc. Right, so you moved to Manchester, and with Professor Nicholas Turner. What I want to ask about that is like how, because people that do postdocs, they usually go on to either become a professor or maybe the job market's not right yet, or maybe you just want to live somewhere else to just do really cool research for a year or two or however long. So, what were some of the like decisions that made you think about doing a postdoc, and why at the University of Manchester with Professor Turner?
Okay, so it's a great question, and it doesn't have a really structured, I thought about all this kind of answer. And I don't think I'm not very good at planning ahead. It's not one of my No, so I just go with what's happening and what feels right. And I was finishing up my PhD at a time when you might have heard of the Celtic Tiger. Back in the 2000s, the economy in Ireland was really booming, and construction industry was going great. There was so much money in this country, probably not real money, but there was lots of it about. But then that crashed, and the economy went
Oh,
completely down. And that was just coinciding with me finishing my PhD research. And of course, at a time when I was ready to look for a job and join the real world. And I was applying for industrial positions in places where I should have been very well qualified. And I was finding that these just weren't available. And I wasn't really ready to leave Dublin. I was happy where I was, but I was almost forced out of the country by the lack of availability within the job market. But of course, in a sense, that was the best.
mm.
In hindsight, of course, that was a created a great opportunity. And actually, one evening, sitting a little bit depressed, probably in my lab in ECD, and just before Christmas, I think I emailed Professor Turner in Manchester because I knew a little bit about the area. I'd seen him give a talk, and I said, would you consider taking me on for a postdoc? Thinking I would never get a reply, but actually, I got a reply the next morning, nice and early in the morning, which was great, to say, let's come and visit us, and let's take it from there. And that's it. It happened all by, didn't plan any of it, but all good decisions in hindsight.
I mean, everything happens for a reason, for sure. I like to think. And I mean,
That's it.
I don't think you have. I don't. I think a lot of graduate students are like, oh man, at towards the end of my PhD, I really have to like be in the job market, or I gotta have everything planned out. It's like, I don't know, like I kind of applied to graduate school on a whim because I wasn't really ready to go to industry yet from undergrad. So I was like, all right, screw it. I guess I'll apply to graduate school, and now here I am. It's working out well. So sometimes you just gotta like just relax and ride the wave. Um, so that's really cool. Now, you had basically spent your entire life in Ireland though, and then you kind of moved over to Manchester. Was there any like, I mean, the Irish and the English is kind of funny, but moving over to Manchester, what were some of the biggest differences you kind of seen between Dublin and like Manchester? Because Manchester is also a huge city.
Yeah. So it was perhaps one of the laziest moves ever, because Manchester is just across the water. And it's okay. It's a great city, actually. I have to say, I love Manchester. I always describe it as my sort of spiritual home. It's a brilliant city, especially when you're young, and there's so much to do there. But it's so like Dublin. It rains probably just as much as it rains in Dublin. And to be honest, like you'll find if you go to the States as well, almost everybody is pretty Irish as well. They've got an Irish heritage. And so it was actually a really welcoming city. It seems like it might not be a big thing. And then perhaps Americans move around a little bit more for university, I guess, than we do. I lived at home till I was 26. And moving to Manchester at 26 was actually quite a big move for me. I moved on my own, didn't know anybody, strange city, no sort of support. So it was kind of a big move, but actually there's such a welcoming people in Manchester. And British people, as you say, are very, very culturally similar to Irish people. So I guess I found it easier because of all that. But it was a fantastic place, and anybody who's keen on research in the UK, Manchester and the University of Manchester is a fantastic place to study, and a great city to live in. So no, I had a great time at Manchester, and I absolutely loved it there.
Yeah, my opinion's biased though, because I obviously like I'm a huge supporter of like football. So everything I know about Manchester basically is from the City fans and United fans, who I can't stand. So I kind of have a biased opinion. I know nothing about their culture. I know nothing about their culture, and I know nothing about like even like the cool science that goes on there. So my opinion is
Well,
just
they have
biased
pretty good
because of
football
for that
team.
reason only.
Yeah,
They do have really good football
they,
teams,
they
which
kind
is
of take.
a shame, really. But yeah. Well, by the way, did you go to any football matches while you're over in? Well, obviously, I'm sure you've been to a few, but well, I think rugby's also huge
Yes,
there
well,
as well. So
I'm a Liverpool supporter,
Um,
so living in Manchester, and I used to play football when I was over there as well. I joined a ladies' football team at one point. But I used to also play five-a-side with all my lab mates, and that was good fun. I broke a few bones. That was less fun, but good exercise and great to kind of be involved in these outside lab things with people. But now, as a Liverpool supporter, actually, when I broke my arm playing five-a-side with my lab mates. I was in goal. That was a mistake, and I saved an absolutely fantastic goal, but it wasn't worth it. I broke my right hand, and unfortunately, I had been
oh my
playing in
God.
a Liverpool jersey, and I had to walk into the Manchester Royal Infirmary Hospital in my Liverpool jersey. I couldn't get it off because my arm was so sore, and I had to sit there quite late at night,
Oh
surrounded
no.
by people looking at me evil stares, to be honest. But yeah, I went to quite a few. Manchester United, I went to the stadium a few times to Old Trafford, which is just such a great. I love football stadiums. Actually, they're just amazing places to be in. And I went to Anfield as well, Liverpool's home ground, quite a few times. And I do miss that. Actually, living in Ireland, it's
Yeah.
not as easy to just miss a football match. So no, that's something I definitely miss.
Yeah, that's I know. Like I know Anfield can be an absolute fortress to go to. You don't want to be playing. Champions League nights at Anfield are just dreadful. Old Trafford also beautiful stadium. Needs I've heard from the fans I've heard they need to do major. The Glazers need to go out. Apparently, I don't know. But anyway, yeah. How is rugby though? Is the big sport in Ireland though? Right? I think not necessarily. I mean,
Yeah,
football I guess is big
yeah.
too, but
I don't follow rugby.
rugby.
Yeah, I don't follow rugby as much. I don't follow anything. I've got two young children, so I just sort of watch, shows that 4-year-olds or seven
Gotta
year
watch
olds
them.
like. So I don't watch a lot. I want to watch. But yeah, rugby is huge in Ireland, and we don't have the strongest football team in the world, but we're a very small country. But it always amazes me how good our rugby team is, and we are so strong at that. And of course,
hmm
a lot of the schools in Ireland, rugby is a huge focus. So we do turn out some really good players, and it's a great sport. It's just my passion has always been soccer. If I was going to watch something, that would be it. But as I said, it's sort of Henry Danger and Peppa Pig is what I watch.
Yeah, well, that's I guess the way it goes. Becoming a mother now, you gotta take those sacrifices now. I don't know. Maybe they'll grow up in like football, so you can kind of get back into it then. Um, but yeah, sorry. So yeah, you were at Manchester doing a postdoc with Professor Turner. Um, yeah, how was that experience overall, and like what were your research interests with Professor Turner?
Yeah, it was a great experience. I was 4 years with Professor Nick Turner and also with Sabine Flitch as well. Sort of who shared a lot of research projects together. So I kind of worked between the two. Brilliant experience. I learned so much, and I mean, it was a very different experience to my group in Dublin. It was also a fantastic experience, but we were a much smaller group in UCD. In Manchester, I mean, between the two groups, there was well in excess of sort of 30 researchers, maybe more. And so that was great. And it really allowed, very multidisciplinary as well. Even within that group, there were real hardcore synthetic chemists, and then there were people who did a lot more of the sort of the structural biology and the molecular biology. So it just gave me an opportunity to learn so much. And that was a great experience. And it certainly taught me a lot. So whilst I had about 18 months in UCD at the start of my PhD, where I would have dabbled in a bit of molecular biology and sort of gene expression, it was still something that I needed to learn a lot about. And I think working in Nick's
Mm.
group gave me that opportunity, and had so many really fantastic supports around me, postdocs who were much better at that than I was, and it was just phenomenal for giving you that. And again, a lot of freedom in that group. So the opportunity to kind of do little projects that I was interested in and develop my own research ideas, which was great.
Yeah, that's really cool. So at the end of your postdoc, then I think. Well, actually, I guess at what point then? Because now obviously you're a professor at back at Dublin, but at what point were you kind of considering moving back to be a professor? Like what were some of the decisions leading into that?
So Yeah, in Manchester when I was doing my postdoc. I mean, I kind of since I did my PhD, in fact, I had thought I would like to be an academic. I love the idea of research being something I did all the time. I didn't really know much about industry, so I thought, I don't really know much about it, and I know I like it in an academic setting, so why not just try stay here? But I understood how difficult that was. It's not easy to just get an academic position, of course. But when I did my postdoc, I was really sure that I would be able to do this and that I could come up with my own research ideas and that it was something I would enjoy. So I then got offered a academic position in a smaller, more teaching focused university, pretty much just across the road from the University of Manchester. So another very lazy move, actually. That was Manchester Metropolitan University. And I got offered that position and I took it. And I only stayed there probably for about 10 months. It was a great place and fantastic colleagues who I still keep in touch with and collaborate with from Manchester Met. But I then, from there, got offered an academic position at the University of Nottingham, which I suppose I couldn't really turn down. It's a really great chemistry department, and I felt that it was a good career move. And so off I went from Manchester down to Nottingham, which is sort of in the Midlands in the United Kingdom, in England.
Yeah, the Midlands of England, moving further away from Dublin. What, how, at the beginning of your professorship at Nottingham, I'm sure things changed between then and now. But what were your first initial projects that you really wanted to take on? Like, what were some of your original ideas?
Yeah, so a lot of, and this is probably quite common, a lot of the ideas that I had in my head were stemming from challenges that I'd had during my postdoc. And I was working with, I started working with cytochrome P450s in Nick's group, and it was a postdoc for the first 2 years, which was sponsored by or part funded by AstraZeneca, so a pharma company. And they were interested actually in asymmetric hydroxylation, so in adding oxygen to molecules selectively, with a view to maybe making metabolites of drugs or making drugs even that might be more active than the parent compound. So they were interested in doing that. So I worked on cytochrome P450s for a couple of years. They were really difficult to work with. You asked that question earlier about enzymes, whether they're easy or difficult to work with. They were super hard to work with. And then I start working on another project on amine synthesis enzymes. So amines are functional groups which are really prevalent in drug molecules and lots of different chemicals. So being able to make those enzymatically and with the sort of the attributes that enzymes offer in terms of selectivity and specificity is really valuable. So I start working with transaminases. And I suppose when I moved to my own independent career, it was looking at what were the challenges that I had in my research to date with those enzymes and could I address those challenges? And the first part of my research, and in a sense it kind of continues, it's evolved of course, but has been in, can we take enzymes and use a methodology which makes it actually easier to do that chemistry, which simplifies that and makes it more sort of plug and play and much easier to use and not such complicated methodology that in a sense would turn a chemist off who didn't have the experience working with these enzymes.
Mm-hmm. Yeah, that's really cool. Yeah, we can definitely hop into this now. So, after a few years at Nottingham, then you got offered the position, come back to Dublin. Actually, how did that opportunity arise? Like, were they just were they looking for professors, and were they like just kind of sent you an invite, or how did that all
So
transpire?
I As far as I remember, again, these things always happen when you're quite happy where you are. And I was really happy in Nottingham. It was a fantastic department, great colleagues. We just bought a house. We were very happy there. And I got
Oof.
an email. I actually think my supervisor, my PhD supervisor, Francesca Paradisi, I think she sent me an email to say, just in case you're interested, dot, 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 with a link with a link to the job advertisement in Dublin. And I remember opening it and thinking, why didn't this come a few years ago before I was all settled here? Anyway, I thought, you know what, I'll just apply and then, let's see what happens. So I did apply. And of course, got offered that position, and I was delighted to have been offered it. But it's a big move, kind of settled in the UK at that stage. I had one child, Harry, who's now nearly 7, and he was sort of settled. I had him when he wasn't that he's 7 now. He was only, was he 3 or 4 when this was coming about? No, he wasn't. He was much younger than that. He wasn't even 2. And we were kind of settled enough there. But look, when these opportunities arise, you have to decide, it mightn't come again. So if it's there now and it's, feels kind of right, you kind of just have the plunge. And I didn't have any family in Nottingham, so I had one child at the time. I now have 2, but I didn't have any family support around me. And I also thought, how am I going to have a second child on the way? Actually, in the very early days of the application to Dublin, I thought, how am I going to survive now with 2 children, trying to hold down a career? And maybe it would be easier being back in Dublin with a little bit of family around me. So that's really what took me back. And I loved UCD. I loved being there in my early years. And I thought, it can't have changed much. It must still be a good place to be.
Yeah. No, I definitely hear that. I mean, I'm originally from Philadelphia, but I do graduate school here in Houston. And Houston's a great city, don't get me wrong, and I love my colleagues and I love my advisor. But I can't imagine like trying to raise a family without having my parents around. I mean, obviously, because my parents want to be grandparents, so they obviously need to be within a reasonable distance, I'd say. So I definitely hear that point of view as well, for sure. How did it feel to kind of come like full circle then? I mean, just a little undergrad coming out of UCD, and now you're back as a professor now. Like, has that like kind of sunk in at all, or is it kind of like no big deal?
It was the oddest experience because, as you say there, I did my undergraduate degree in UCD and then I did my PhD there. So I knew all of the academic staff very well from my studies, but I knew them as my professors, as people who lectured to me, or as the PIs and research groups. So coming back, actually, I don't mind interviews. I know some people get really nervous for interviews, but I actually quite like interviews. I just enjoy them. But I hated that interview for UCD because, normally you're in a room with people you don't really know very well. So, they don't really know much about you, and the dynamic is totally different. But when you're in a room with people who know you really well, and who know that, you screwed up this when you're in your undergraduate labs and you weren't very good at that, it's a terrible situation to be in. And so that was it was a lovely interview, but I hated it. Actually, it was the one interview I didn't really enjoy. And then when I did get offered that job, and when I came back to UCD as then a colleague to these academics that I had that had been my professors, that was a very strange dynamic. And I think certainly for some of the more senior colleagues, it's actually quite difficult not to refer to them as Professor such and such. Just calling by their first name was even a challenge. But it's a great place to work, UCD, and the School of Chemistry is just such a nice place to be, and everybody's been great and very welcoming. And actually, you'll find that, and this might be true in other universities as well, but Irish people tend to go away and then they often want to come home. So I also see now that a lot of people who I knew as a student are starting now to come, or to try and come back to UCD as well. So there's actually a few colleagues even now that I would have worked with closely, or maybe not so closely, during my time as a student as well. So it's not that unusual a situation.
No, I mean, especially like when I can only really imagine like a place, a city like Dublin, where I'm sure it's like it's like the city vibes, but also like there's something about like the hominess of it. Like there's something like really that's like just feels nice, like you're at home. So I can only imagine like when you don't have that anymore, like you want to come back for sure. I definitely hear that. So so now you're back at Dublin, doing really cool research. You do a lot of stuff, making enzymes. I know you work a lot with like cytochrome P450s, and capturing that to do synthesis, but also like making chiral amines. And so I guess we kind of touched on a little bit earlier, but I kind of want to come back to a little bit, is that you mentioned a lot of your research interests now kind of came out of the struggles of your postdoc experience, or maybe your PhD experience. So, I guess we could start with maybe like a broad overview of like your research interests, and then we can kind of we'll take a dive into specifics then.
Yeah, sounds good. So, yeah, we're really chemists. I suppose I often, if I was to describe our research group or the type of research we do, most of my group, including myself, are chemists. Now, I have had group members who've come through other degree pathways as well, and that's important because we do quite a bit of sort of research involving molecular biology and things like that. But in general, we're just chemists trying to make molecules, and they could be pharmaceuticals or sort of building block chemicals or even bulk chemicals. But using enzymes to do that chemistry, as I guess a lot of people will know, can be really efficient, because if you're trying to do something using traditional synthetic chemistry, like you talked about amine synthesis there, you've got a lot of challenges with selectivity and specificity. Whereas enzymes in nature, they have to be excellent at their job. So they have to be able to take a molecule. You remember, you're in a cellular environment and you've got loads of biomolecules, thousands of biomolecules within the cell. So the enzyme that's in there doing the chemistry has to be able to pick out from those potentially hundreds or thousands of molecules in that environment the correct molecule that it's going to work on. And then even within that molecule, it's got to pick out the correct part of that molecule that it's going to install the amine or do the hydroxylation reaction on. And so it's that specificity that nature has spent billions of years evolving and designing into these macromolecules, these catalysts, that we try to take advantage of as chemists. But really, we're just, and I always sort of describe this to students who are thinking about joining our lab, we're not that interested in the enzymes themselves. We're interested in what we can do with them. So it doesn't matter where we get our enzyme from. It doesn't matter, once it does the job, that's what we need to do. And sometimes they need to be engineered or evolved in order to do the job that we need them to do.
Mm. Yeah, so I know you're generally interested in amines, but how does one listen, I don't know anything about like the libraries of enzymes there are, but how do you just pick out which enzymes are like that may work? Like, I don't know, do you have like a initial screens, or like I don't, how does that kind of process begin?
Yeah, so it really depends on what you're trying to make. So even with just sticking with amine synthesis, which is really the bulk of the type of research that we do, and even within there, you've lots of different enzymes that can install an amine selectively. So it really depends on what sort of an amine you want. You want a primary, secondary, tertiary, what you're trying to make. But also it depends on what you can do with the enzyme. So transaminases, for example, are pretty good enzymes. They can allow you to make primary amines, but their substrate scope is quite broad. And what I mean by that is that you can actually they take carbonyls, aldehydes or ketones, and they let's just take those two examples, and they can convert those into chiral amines if you're starting with a ketone, for example. And their substrate scope is quite broad, which means that they can actually accept quite a broad range of ketones or of aldehydes. And that's not true for all enzymes. So you might be more restricted if you wanted to maybe take a different type of enzyme to try and make that amine. You might be more restricted with the type of substrates that you could access. So those are some of the decisions you might make. But in terms of just a specific enzyme, say you're just trying to decide on what enzyme you would use for a particular job, and you mentioned screening in there, that can be a really big part of what we do. If we want to make a particular amine, maybe it's very valuable, it's very difficult to make using more traditional synthetic approaches, we might have to screen libraries of transaminases, for example, and that could be anywhere from 10, 20 up to hundreds, thousands, or even hundreds of thousands sometimes, to see which enzyme is going to do that job for us.
Okay. Now, a couple questions on that. So I assume you have to purchase these enzymes though, right? Like I don't know how easy enzyme synthesis is. Like I don't really know. I'm sure you can make some enzymes, but and then so purchasing enzymes, but also how, like, let's say, like, if you're, for example, if you want to make like I don't know, in one of your papers you make like cyclic amines using one of those transfer aminoaces. Like, what is that? How what does like the procedure look like? Like how does one just like put in the enzyme? You need to keep it like really cold or something like that. Like how does that kind of work then?
Okay, so great questions. So there's two answers in a sense to the first one, or at least two broad answers. You can just buy enzymes if they're commercially available, and there are loads of enzymes, loads of different classes of enzymes that do lots of different things that are commercially available. But say you wanted to make an amine with a transaminase or with a cytochrome P450 or something like that, you could purchase it online like you would buy any chemical that your group would be interested in buying, or any palladium catalyst or any ligand that you're interested in buying commercially. You can buy an enzyme, and it comes as it often comes, not always, it can come as like a powder, and you wouldn't even know that it was an enzyme. You just tip it into reaction and off you go. But actually, the beautiful thing about using enzymes, you don't have to do that. So if you go to Sigma Aldrich, or one of the big companies that we get our enzymes off actually is Codexis, and they produce enzymes. They actually produce new enzymes, and then they package them, and they sell those as well. But we would use a lot of their enzymes for our methodology. So once you buy it and use it, it's gone, like any chemical. And then you've got to go buy it again. And enzymes can be quite expensive. However, the beautiful thing about enzymes is that if you can, you know, it's a protein that you're using. But of course, that protein has come from a gene. So if you can get your hands on the gene that corresponds to that protein, well, you never need to buy the enzyme again. So all you need to do is buy yourself, for example, some E. coli cells that will produce that protein for you. Stick your gene, and it's a complicated enough procedure, but it's really well established. Put your gene into that bacterial cell, grow the cells, and ask it to produce, use some methodology to get those cells to produce potentially bucketfuls of protein for you. And then you freeze the cells, and you 5 years later pull them out and do it again. So you actually never need to purchase a protein if you have access to the gene that corresponds to that protein. So I mean, that's
really
really
cool.
powerful. If you
Really
say
cool.
to us, it's really cool, yeah. You can't do that with lots of, you got to go buy all your metal catalysts and things like that. You can't get a bacteria to produce these things for you. So that is really powerful. And then your second question, how these are used. Well, the first way, when you just buy it from a company, well then, okay, you typically do your enzyme reactions in an aqueous environment, like in water. So it might be some type of a buffered system. But again, it's more straightforward than you think. It really is. You've got your aqueous system, you got your substrate in there, you spoon in your enzyme, and off you go. If you're using genes that you've produced yourself, you might purify the protein, but you might actually just choose to throw in your bacterial cells. So if you imagine a cell that's produced this protein for you, it happens in our bodies all the time. Biomolecules slip in and out of the cell, and the chemistry takes place when it gets into the cell. The product then leaves the cell. Why not just do whole cell catalysis as well? So we would have done a bit of that in our research group as well, where we don't even bother purifying the protein, and we just use a bacterial cell, and we do all our chemistry relying on the substrate going in and the product coming out.
That's really cool. Now, I assume this might be obviously could be like enzyme dependent, and this could also be an ongoing area of research in your lab. I don't know, but understanding, let's say, the enantioselectivity or stereoselectivity in these reactions, is that something that you already know, or is that something ongoing? And how does that like work?
Yeah. So firstly, you don't always, even if you think you have an enzyme that is going to give you an S product, it's often substrate dependent. So I suppose if we start maybe about how that works, enzymes are big macromolecules. They have this active site usually buried deep within that protein. And that active site is, of course, three-dimensional. And the nature of the way enzymes work is they bind the substrate. And of course, their specificity, and the thing that I mentioned earlier, that nature has to do these things extremely selectively. So it has to bind that molecule, usually in one very specific orientation, to allow catalysis to happen. And that might mean binding it so that you can have a hydride delivery, or so that it's positioned perfectly for whatever that mechanism for that catalysis to take place. So that three-dimensional shape ensures that that molecule binds in a particular orientation. And so when, for example, you've got a hydride delivery or water, whatever that chemistry is, that's what gives you your selectivity, the fact that it's bound in one particular way. And that's, of course, really powerful. But if you think about that, those very subtle binding interactions that take place, just because of amino acid residues that are in this binding pocket, these really subtle interactions, they're so powerful. The ability of these amino acid hydrogen bonding interactions and van der Waals interactions, and these complex interactions that you talk about with enzymes, they're very strong because there are so many of them. But if you disrupt that very delicate balance of interactions by maybe just changing the substrate from what nature is typically used to working with, so the natural substrate, and you might just change that substrate very subtly, so give it a substrate that nature doesn't use, that can disrupt this very delicate balance of interactions, and all of a sudden that doesn't bind anymore, or it binds in a different orientation, and therefore the selectivity can even change. So when people talk about, and we talk about them ourselves, an S selective or an R selective P450 or transaminase or whatever it might be, it's often substrate dependent. So you do have to be a little bit careful. That can change depending on what you're feeding the enzyme.
Mm. And then one other question I had is then how robust like are these enzymes? Meaning like if you put in substrates that, whether they, let's say either like they don't bind well or they don't give you good reactivity, could you like still recover the enzyme? I guess it's kind of a general question, I guess. Even if it does work, right, can you kind of recover these enzymes, I guess, is ultimately really my question. Or do you kind of see degradation?
So some enzymes are much more stable than others. But if you really want to recover your enzyme, and if you think about enzymes in a sort of green, sustainable sort of sense, or if you're we work on very small scale, and recovery is often not that important to us. But if you're trying to do something on a ton scale in industry, and you've spent you've spent quite a bit of money, it's cost quite a bit to produce this protein, kind of it just throwing it away at the end isn't really sensible, cost effective, or sustainable. And so actually immobilization is one of the most effective ways to recover your enzymes. That involves taking an enzyme and immobilizing it or binding it onto some type of a support. And there's a whole range of different supports depending on the enzyme. And usually they're sort of beads or something like that. And so what you can actually do after your reaction is, in crude terms, filter off your reaction, maybe wash the beads and recover them. And then you've got your enzyme bound to these beads that you can throw into your next reaction. And of course, there's lots of ways to do that, but that can work really very well, and it can affect the activity of the protein, but that effect can be sometimes enhancing the activity. So it really just depends on the protein.
That's fair. As we kind of begin to wrap up, because I know you got to go pick up your kids, but I know you recently, what initially got my attention was you recently published a little paper on capturing, using cytochrome P450s to do, to do synthesis, which is really cool. And the brief things that I know about cytochrome P450 enzymes, and we kind of mentioned it earlier. I mean, they are they're very good at what to do inside the body, but obviously if you take them outside the body, one is I think heme groups are actually toxic. So they're quite and iron is, I work in organometallic iron, but iron is also not exactly the most easiest metal to work with. But I guess speaking to that publication actually a little bit, maybe breaking down, the utility of cytochrome P450s to make ketones, and why this is like a really awesome area.
Yeah, so you're not talking of, you're probably referring to a news and views that I wrote on an absolutely beautiful paper for using these enzymes to make ketones. We actually, I'll tell you the reason we don't work with cytochrome P450 so much ourselves anymore, and it's not because they're actually, I'll answer that question in terms of their utility and their usefulness. They're actually one of probably the most powerful enzymes that I can think of. It's the holy grail of synthesis, to be able to take an unactivated carbon chain, for example, and oxidize it selectively, because that's incredibly difficult to do. You're asking an enzyme to take something that isn't particularly reactive and to pick out a CH bond somewhere on this carbon chain and oxidize it. And that, I think you're referring to a News and Views on some beautiful work published in, I think it was Nature Chemical Biology. The reason we don't work that much with cytochrome P450s ourselves anymore is they are so, I think my heart got broken during my postdoc because they're very difficult enzymes, or they can be very difficult enzymes to work with. But in terms of, I think if you asked me what maybe the most powerful enzyme in terms of synthetic capabilities are, it probably would be cytochrome P450. So we might come back to them ourselves. I worked a lot with them during my postdoc. We might come back to them one day when I forgive them for all the hell they put me through in my early years. But we're very lazy these days, and we tend to work a lot with amine synthesis enzymes, which also are sometimes challenging to work with, but maybe a little bit more forgiving than some of the SIPs.
Yeah, what is it kind of well understood why let's say like synthetically cytochrome P450s are difficult?
There's quite a few reasons for that. It really depends on the source of the enzyme, for starters, how difficult the protein itself is to work with. And then I guess some of the challenges that exist with P450, certainly the ones that stick out to me and the problems that I met with them were their promiscuity. So again, if you think about what a cytochrome P450's role in nature is, a lot of these enzymes are in your liver. So in mammals, these enzymes are in your liver, and they have lots of different functions, and they're there to hydroxylate, make things more, so add oxygen to molecules to make them more water soluble so that they can be excreted. So a lot of metabolizing enzymes, and of course they're in bacteria and other systems as well, but their role is to hydroxylate. And they can often do that not only on a huge number of molecules, but within the same molecule, they can actually hydroxylate in a number of different positions and even do sort of different chemistry on the same molecule. And so what I would have found when I worked with these enzymes myself quite a bit when I was in Manchester was that instead of, we're used to working with an enzyme and saying you feed in a substrate, you put in your enzyme, it does a reaction, and you get your product out the other end. But sometimes with cytochrome P450s, you get lots of products out the other end because the enzyme sort of just hydroxylate. And not always. There are some very specific ones. And of course, that's where we probably won't cover this today, but that's where in engineering enzymes to enhance their specificity comes in. You could take a cytochrome P450 that actually is quite promiscuous in terms of its ability to hydroxylate in lots of different positions and turn that into a more selective catalyst. And that's another beautiful aspect of enzymes, is that you can actually tailor them, again, working on a DNA level, working with the gene, and you can engineer them to be more specific, to be more tolerant in organic solvents, to withstand high temperatures or cold temperatures. They're really very tunable. And we haven't touched much on that today, but that's perhaps one of the most powerful things with enzymes, is that they're tunable.
Yeah. I guess I should mention that. Cytochrome P450s, and you can definitely touch on this. They are responsible basically when you take, let's say, if you take a pharmaceutical drug, like they're responsible for metabolizing it basically and trying to get it out of your system. It's basically your body's self-defense system for alien molecules that enter your body. Generally speaking, that's what they do. And yeah, now that you say it, it does make sense that like if like a molecule entered your body, the cytochrome P450s would start hydroxylating things just to so you could secrete it through urine. And so that kind of makes sense. And I could definitely see why there's not much selectivity, or like you have to get very specific when you're talking about like cytochrome P450s. Like it gets very specific. I can see why that would be very difficult. And how you say promiscuous, that is kind of funny way to describe it.
Yeah, but that can be a good thing, remember, because,
right.
Because I mentioned that you can tailor enzymes and mutate them and tune their substrate scope and direct it down whatever route you want. And really, that's very powerful methodology, directed evolution. it's not the worst thing, because if you have a cytochrome P450 that's able to do lots of different jobs, well, that's good, because you have a template, a starting protein template that is potentially the sort of the mother that can be turned into lots of different catalysts for lots of different specific functions. So it's not good when you're trying to work with that parent protein and produce one product, but it's kind of a great starting point for engineering enzymes to do lots of different chemistries. That promiscuity, as I talked about, can actually be quite valuable when you come to engineer the protein.
Mm. Well, Professor O'Reilly, I want to thank you so much for coming on to the show today. It was an absolute pleasure talking with you. And I look forward to seeing more about your enzymes and chemical synthesis coming forward. It's very exciting. It's a very exciting time to be working, I think, in enzymes. It definitely makes a new paradigm for chemical synthesis. So it's super, super fun right now. And there's so many enzymes, there's really never ending. I feel like never ending research in this area. So that's super good for you.
Thanks, Aidan. It's been great to talk to you, and you have an open invitation to UCD for a pint of Guinness anytime you want.
I will surely be using that within years to come. So thank you again, everyone, for listening, and I'll see you in the next episode.